All right, everybody, we are starting chapter 10 today, DNA and synthesis of proteins. On slide, I know you can do it. You can change. All right, this module has 4 parts. First is DNA structure, second is DNA replication, and we're going to do those 2 parts today. Third is protein synthesis, and fourth is mutation. When we say a gene codes for a protein, that means that it encodes the information necessary to make that protein. DNA is basically a code, and when the cell decodes that information, it can use that information to make it. So first, let's talk about why DNA is so very important. Well, because of its relationship to proteins. You may remember this chart that we drew in class probably about a month ago at this point. But DNA dictates which proteins a living thing is going to make, and proteins do basically everything in that thing, that cell, that plant, or that animal. This diagram focuses on human systems, but the principle holds true for all living things. Different portions of DNA code for different proteins. So for each of these things, there is a specific protein, a specific piece of DNA that the cell uses to make these things, or not make them if the DNA isn't there. I was thinking, why do I love DNA so much? And this is why, because DNA forms the very basis of who we are and how we function through proteins. So here we're gonna have a little philosophy connection. DNA, chapter 10 is my favorite chapter out of all the chapters. DNA is so fascinating to me, and here's why. First, let's talk about sex and gender. So most people think of themselves as either male or female, and that affects a lot of what they do and a lot of how they interact with other people throughout their entire lives. And we're most commonly taught that if you have 2 X chromosomes, you're female. If you have an X and a Y, you're male. So your DNA dictates this part of your identity. Well, that holds true for most people. In fact, this is called the XY sex determination system. It's the system used in humans. In turtles, sex determination doesn't have to do with chromosomes. It has to do with temperature. Clownfish have the ability to change between male and female throughout their lives. The sex determination schematic, the way it works, is different for different species. But back to humans. So by definition, sex is the physical, biological result of your genetics. It's what your genitals are, your reproductive organism. Females have ovaries and a uterus, and males have testes. The word gender refers to whether a person considers themselves to be male or female or possibly something else. 2 X chromosomes, in terms of gender, doesn't always create a woman, and an X and a Y doesn't always mean you're a man. And this might sound silly at first, or like it defies some kind of logic, but hold on just a second. What actually causes male genitalia to form is the SRY gene. That's found on the Y chromosome, and the SRY gene creates the SRY protein, or the sex-determining region Y protein. When developing in the womb, we're all female by default. Every man has the genes for a uterus and for a fallopian tube. But the SRY protein, it attaches to those genes and inactivates them. And here's an image, the DNA right here. Where's my mouse? This is part of the gene for the uterus and fallopian tubes, and you can see the SRY protein right here attaching itself to that DNA, binding to that DNA, and preventing the cell from accessing the information on that DNA, and that's how it's inactivating those genes. So this is why we're taught that someone with 2 X chromosomes is female and X and a Y is male, because the SRY gene is in the Y chromosome, and males have that. Remember, we're still talking about sex here, the genitalia. We're not talking about gender, which is whether someone considers themselves to be male or female, or perhaps even something else. But occasionally, things go differently. When eggs and sperm are being made through the process of meiosis, chromosomes, they exchange genetic information through crossing over. And normally, only chromosomes that carry the same genes will exchange genetic information with each other. but sometimes different chromosomes will exchange genes that they're not supposed to exchange, such as between the X and the Y chromosome here. And in some cases, the Y chromosome will give the SRY gene to the X chromosome through crossing over like this. So now we've got an X chromosome that has been altered, and it has the SRY gene on it. And this could lead to a fetus with no Y chromosome, but they have the SRY gene anyway. This condition is called 46XX. A person with two X chromosomes will develop male reproductive organs. So their genotype says they're female, but they have male reproductive organisms. So are they male or female? What is their identity? Someone with this DNA might develop as a male, but if they have their DNA tested halfway through their life, they're going to find out they have no Y chromosome. Sometimes there might be a mutation in the SRY gene, and depending on the mutation, maybe the SRY protein won't be made at all, or maybe it will be made but it doesn't work. A fetus without functional SRY protein will develop a uterus and fallopian tubes, even though it has a Y chromosome. And often these non-functional organs or ovaries will be formed called streak ovaries, and they can become cancerous, so they must be removed. And if that happens, the person has to take estrogen if they want to develop female secondary sex characteristics, such as breasts. In this case, the person might grow up female, but if they have their DNA tested, they have a Y chromosome. So are they male or are they female? What should their identity be? Here we have an individual with a non-functioning SRY gene. This could lead to Swyer syndrome, and that affects about 1 in every 80,000 births. You can see that this person has a somewhat male, or maybe even ambiguous appearance until high school, and in high school they began taking estrogen, which led to a more female appearance. So should this person consider themselves male or female? The question is, should DNA determine their identity? The mutation in the SRY gene causes an SRY protein that functions a little bit. It doesn't do the whole job, but it isn't completely absent. That's called partial gonadal dysgenesis, where individuals have genitals that aren't clearly male or female. It may also cause other abnormalities in the reproductive organs. So, should this person consider themselves male or female? Should their DNA determine their identity and how they want to conduct their life? I imagine that a lot of you are probably wondering about hermaphrodites at this point. So I was researching this and I found a fantastic article on Britannica. There is honestly no better way to put things than the way this article does. I thought about what I would say to summarize the article, and then I realized that the article was just a perfect summary. So if you're interested in learning about hermaphrodites, you can go here and read the first 2 paragraphs. The rest of the article is interesting too, but the first 2 paragraphs are what really talk about hermaphroditism and what it is biologically. So back to our first slide here, there isn't always a direct correlation between sex and gender. So if the genes don't match whether a person is male or female, then who should decide whether this person lives their life as a woman or a man? Maybe they don't feel right being either one completely. And that begs the question of whether a person's DNA should cause them to live as male or female at all. If they have a quote-unquote normal genotype, normal X and Y chromosomes, does that automatically mean they have to live their life and behave in a specific way? And this is what transgender is. When somebody is born with DNA that does not match who they feel they truly are. A person might have 2 X chromosomes, whether the chromosomes are completely functional or not, and still feel that they're male and want to live their life as male because they feel that their DNA doesn't dictate their identity, who they are. And the same is true for the opposite. A person might be born with X and Y chromosomes and still identify as female. And this leads into our next point, which is that some genes affect brain functions, and that in turn affects how people interact with their environment. Multiple genes work together to shape one characteristic. They may have other effects as well, and of course environment plays a role in determining a person's personality. These are just a few of the genes and the proteins that are associated with a few personality traits. There have been genes and proteins found that are associated with things like excitement seeking, and that is the protein called ctNNA2. With openness, we have proteins RASA1 and PTPRD. Conscientiousness, agreeableness, neuroticism, there's a lot there. Extroversion, there's a lot there. So I want to be clear that these things don't cause these characteristics, these personality traits, but the existence of them has been correlated with these personality characteristics. The question is, if you have this DNA, does that define who you are and how you live your life as a person? DNA also creates proteins that in turn create neurotransmitters. Neurotransmitters play a huge role in personality and in overall mood. Serotonin is involved in happiness. If you don't have enough serotonin, you're going to become depressed because your brain can't send enough of the nerve signals that are necessary to feel happiness. Dopamine is involved in memory and motivation as well as in mood. Serotonin and dopamine are not proteins, but proteins are needed to make them, and these are the proteins right here. So you can see, for example, tryptophan is turned by this protein, tryptophan hydroxylase, into 5-hydroxytryptophan, and then that, in turn, goes through this protein, aromatic L-amino acid decarboxylase, and then you finally have serotonin. And this process is similar for dopamine. Antidepressant medication doesn't change your DNA. It just helps your body conserve your existing neurotransmitters. So if the DNA you have, the genes you have, don't allow you to produce enough of these things, then you can take supplementary serotonin or supplementary dopamine to help compensate for that, for what your DNA isn't allowing your body to do. Anyway, enough about that. Let's move on. The purpose of DNA is that it stores directions for making proteins, and it's called the hereditary material because it is passed from parent to child. Remember that the protein monomer is an amino acid, and the shape, or the structure, dictates the function. A gene on each long DNA molecule, there's many different genes. Each one codes for a different protein. Here's a graphic showing how the genes are found all together on the same chromosome, and each one codes for a different protein, and those proteins could do any number of different functions, as we discussed before. All right, an overview of Chapter 10. Let's say you want to make some food, and maybe a friend has a great recipe for pizza, and you want to make that pizza. Get off the counter. First, you get a copy of that recipe. Well, why? Because you don't want to take the recipe. Then your friend wouldn't have one. And if you have your own copy, you can take it anywhere you want. And then using the information on that copy, you, the chef, you make the pizza. So it's a transfer of information between different forms. That's what's going on here. And the central dogma of biology shows the information, the flow of information from DNA to protein. It's just a transfer of information in the same thing. And this is what Chapter 10 is all about. And you're getting a little preview of what we're going to be talking about this week and next week. A cell can do two things with its DNA. First of all, it can use the existing DNA as a template to make more DNA. That's called DNA replication. Also DNA synthesis, and that's what we're going to talk about today. Or the cell can use its DNA to make proteins. This is called protein synthesis, and it has two steps. The first step is transcription, in which DNA is used to make RNA. It doesn't turn into RNA, it's just used to make RNA. And then the second step is translation, in which RNA is used to make a protein. And each of these little individual circles represents an amino acid, and each letter stands for a different amino acid. So I want to be clear that in translation as well, the RNA is not turned into a protein. It is used to make a protein. Oh, gosh, I'm almost out of time. Okay, just kidding. Move on to part 2.